It's my great pleasure to be able to introduce Dr. Are you already doctor? Obviously. Yep. Dr. Alec Christie to everyone here at CBCS. So Alec received his bachelor in marine biology from the University of St Andrews in the UK in 2017. He then moved to the University of Cambridge to complete a PhD in zoology, which is about evidence-based conservation. And he finished earlier this year his PhD. And in October, so in 3 days, he will begin an extremely prestigious junior research fellowship at Downing College, Cambridge, at Cambridge, working with the Cambridge Conservation Initiative to advance evidence-based conservation efforts. So I met Alec for the first time when he started his PhD about 2-3 years ago, and it was just amazing to see over the past 2-3 years what he has revealed and achieved to understand how we should collect and use scientific evidence for evidence-based conservation. And obviously, he has been working with the Conservation Evidence Project, which is a really exciting and interesting, important initiative for biodiversity conservation. So I believe he will be talking about some of these exciting works. So please enjoy his talk. So Alec, if you're ready, over to you, and please start sharing your screen.
Okay, brilliant. Thanks for those kind words, Tatsuya. Yeah, I'll just start sharing. Okay.
And so I'm sorry. Sorry, as always, if you have any questions, please post it in the chat box, or after his talk, please. I'm sure you can speak out. Yeah, sorry.
That's all right. Can you hear me okay? Yeah.
Yes.
Great. Yeah, I just changed the microphone settings because I realized they were set up wrong before, but anyway, that's good. So yeah, thank you. Thanks for inviting me, Tatsuya, to give this talk today. Very excited to be here. So yeah, today I want to talk to you a bit about my PhD thesis and kind of the research I did alongside that. So kind of what I did, what I found, and what my sort of research suggests we should be doing in conservation to inform our decision making with scientific evidence. So I'd had a little slide about me, but Tatsuya did such a good job of introducing me that I probably can skip over this. I'll just show you this video though that I quite like to show because it's quite a good fun starting point. This is when I did some field work in Antarctica and I was trying to film a podcast and these penguins completely upstaged me in the background. So this is part of my marine biology course at St Andrews. Yeah, they got into a bit of a fight. So yeah, something I always find quite funny to show people. Yeah, so like Tatsuya said, I did marine biology at St Andrews. Then I did my PhD at Cambridge with Bill Sutherland, and Tatsuya was my co-supervisor along with Phil Martin. So yeah, it's really useful discussions and advice he gave me to help complete my PhD. And then my postdoctoral work, again, as Tatsuya said, I did that from March this year, and I worked at an initiative called BioRisk, which is looking at lots of biological threats and risks that affect the world. The main one in which that I was looking at was invasive species and how we inform decision makers on how to eradicate and control invasive species. And then, as Tatsuya said, I've got an exciting fellowship that's starting in a few days' time, so I can continue all of this work. So that's really exciting. So just to give you an outline of what I'm going to talk about here today, I'll first give you a bit of background about the research I've been doing. And then the way I've sort of structured it, and this is the way I structured the thesis, was to look at different gaps and biases in the evidence for conservation. So the first is biases within studies, and the second is biases between studies. And once I've talked about those, I'll go on to sort of to mention about the kind of the gaps and biases and sorry, how we can address those gaps and biases and promote more evidence use in conservation. And then I'll round that up for you. So as a bit of background to what I've been doing, I quite like putting it now in the context of the COVID-19 pandemic, because I think that's sadly, but in quite a stark way, it's shown the power of scientific evidence to save lives. And at the same time, of course, we know there's this continuing rapid decline of biodiversity that hasn't gone away. And we also know from a lot of research that's been done before that it's a really poor use of scientific evidence in conservation. And so something that particularly people in my group, but also in other groups, have wondered for quite a while is, why don't we follow this kind of the evidence-based medicine approach to better use scientific evidence, and instead of looking at how to treat patients, can we look at how to treat our planet and save wildlife? And so in the early 2000s, there were kind of a lot of calls in conservation to do more evidence-based conservation, to follow the example of evidence-based medicine. So doing things like systematic reviews and collating evidence in a more systematic and rigorous way to then inform decisions. And so what I was kind of interested in with my PhD is what's happened since then to the scientific evidence base. Has it actually changed? Have we actually built up a good evidence base to inform decision makers in conservation? And then also, what do we need then to keep going along this path? So we've started off trying to be more evidence based. What do we need to do in the future to kind of make sure that continues, and we can really make sure we're following maybe not following the example of medicine, but at least adapting it and coming up with our own approach that kind of is just as successful. So let's now look at the gaps and biases in the evidence for conservation. That was the title of my thesis, was determining the biases and consistencies in the evidence for conservation, and there it is. So again, like I said, I focused on I tried to structure it into two different biases. So biases within studies. So by this, what I mean is kind of those methodological biases, things affecting the internal validity of studies. So mostly study design, and that's what I really focused on. And so I wanted to look at, first of all, how much bias is associated with different sort of experimental and observational study designs, and how often are each of those used. So kind of, looking at those two angles. And then I also obviously wanted to look at biases between studies. And I'm just going to, in this talk, break it down into two different types, because I don't think I have time to talk about all the different ones I looked at. But the main ones I think are geographic and taxonomic, and they're kind of some of the most interesting. So the geographic biases are kind of answering, where are we testing conservation actions in the world? And the second, taxonomic, is, what are we testing these conservation actions on? So let's first look at those within study biases. And so when I talk about study design, what I mean is a definition, a nice simple one that I like, is an organized approach to collecting data. And this is usually some kind of experimental or observational study design. So either like a randomized experiment or some other approach, like a before-after design, where you compare the state of something before and after something has happened. And study design is really a good way, or one way at least, to estimate kind of the quality and the reliability of a scientific study. And that was kind of what I was interested in. And to kind of break it down and classify different study designs, I came up with these sort of three main components that can sort of define a study design. So the first of all is randomization. Obviously, that's kind of one of the most fundamental differences between certain study designs. So you have randomized experiments, and you can have non-randomized or quasi-experimental designs that kind of, they're not randomized, but maybe they seek to try and minimize bias in a way that tries to draw upon the principles of randomization. Then there's controls. So having a control is quite a fundamental part of doing an experiment or doing a study design. And there's also this idea of sampling before and after some impact or intervention has occurred. So if you're interested in, for example, here, the effect of ploughing a field on the numbers of birds in different fields, then you could sample before and after that occurs. And so using those three components, you can define and classify some broad types of study design. And then what I was interested in was testing how reliable and prevalent each one was. So to kind of go through the study designs that I looked at, I've got this quick diagram here. So these are non-randomized designs. So what I mean by that is that they don't randomly allocate experimental units or samples to impact and control groups. So it's not some kind of random algorithm that assigns them. It's done in a non-random way. And the first design, which is extremely basic, is this after design. And the way I've got this diagram set up is, imagine that there's an intervention, like one action that you want to carry out, which is ploughing a field. And so you can sample in fields that have ploughing and fields that don't have ploughing. So that's the top and bottom rows. And you can sample before that ploughing occurs and after it occurs. And that's the two columns. And so with this after design, all you're doing is over time, over several years, you're just looking at how the population of these birds change in the fields over time. So it's kind of a time series. It's not really telling you very much about what the ploughing has done. So I'd probably call it monitoring. And you can do this design called the before after design, which as its name suggests, you sample. You kind of look at the change from before to after ploughing has occurred in some fields. You can also do a control impact. So you can compare your impact and control groups. So for ploughed and unploughed fields after the ploughing has occurred. And you can also do a more complicated one, which is a before after control impact, where you look at the change or the difference between impact and control groups before versus after. So the idea of this is that you take into account the state of the fields before any ploughing has occurred in both the impact and control groups, and that helps you better estimate the difference between impact and control after the ploughing has occurred. And that's particularly important because this is a non-random design. So as I move on here to the randomized design, so a classical randomized controlled trial that's often used in medicine. What this does is, because it randomly allocates groups, so your experimental units are randomly allocated to impact and control groups, you know that the biases, effectively the biases before that might have existed, the differences that might have existed before the ploughing has occurred, so between your impact and control groups, is effectively will average out to zero. So it just becomes noise. So it is quite an effective way of removing bias. And there's also even more complicated design that you can undertake, which is essentially just, both of these are just versions of the designs we saw before, but with randomization included. So I won't spend too much time on this, but essentially there's several different designs that I was able to classify. And I was then interested in how much bias was associated with each of these. And one way you can get at the bias that these different designs have is through this idea of design degradation, which Tatsuya helped me with quite a lot, to kind of, as a way of comparing the results you get from different study designs, but within the same data set. So bear with me. So what the idea of this is that you have this data set here. So you've got a before after control impact data set. So you've got 4 components. You've got the before and after periods. And oops, sorry, my phone's going off. I know it's my watch, that's why. Sorry. So you've got these before and after periods, and you've also got these impact and control groups. You've effectively got 4 components, so 4 parts of this grid here. And what you can do is you can mimic the use of simpler designs by removing some of these components. So here we've got enough data to do a before after control impact design analysis, but you could remove your control data and you could then run a before after comparison, and you could see what the results would be from that. And similarly, you could remove the data that you have from the before period, so before ploughing, and you could do a control impact comparison. And the point is here that you're using the same data set, but you're just using different parts of it. So you're effectively comparing these study designs within the same study. So you don't have issues with comparing across different studies that may have been conducted in slightly different ways or in different time periods or on different species. So these are really nice direct comparisons that we can make. And so I did that kind of empirical research using that design degradation technique, and I also did some simulations. And basically from both those studies, what I found was that clearly some study designs are really poor and some are much, much better. And if I skip through here, so on the left, I did some simulations. So I looked at kind of the ability of a study design to correctly predict or get within a certain range of an estimate, a simulated sort of estimate or true effect. And I sort of used different metrics like this one. So to look at whether the 95% confidence intervals overlapped with the true effect and whether they were within a certain magnitude of that true effect. And on the right, I also ran a sort of more complex hierarchical Bayesian model that looked at the empirical data that I had from before. So from the design degradation sort of experiment or analysis that I did using real data, I was able to fit a model that kind of partitioned bias and variance into different components, and it allowed me to essentially estimate the bias associated with each design. So I don't have time to go into that just now, but essentially what both of them showed was that if you use a randomized controlled design or a before after control impact design, they typically suffered from a lot less bias than control impact or before after designs, so the red versus the blue here. And so that's kind of quite a big recommendation there. To use these more complex but also more rigorous designs because they minimize bias. So my next question was then, okay, so we know some designs are clearly this, we can quantify how much better certain designs are than others. How often are they used? And so I looked at, first of all, the conservation evidence database. And I imagine Tatsuya might have shown this to some of you before, but the idea is that it's collated lots of scientific studies that have tested the effects of some kind of conservation action or intervention. So it's a great online resource, and it covers lots of different fields, lots of different taxa and habitats, and it's essentially got loads and loads of studies that I was able to extract the study design from, and then get an idea of how often these study designs are used. And I also wanted to compare it to another field. So I looked at the Campbell collaboration reviews in social science. So that gave me a nice comparison from another field. And so probably unsurprisingly, I found that the less biased designs that I'd found before were used a lot less than we'd hoped. And so in conservation, these kind of randomized and controlled and before after control impact designs are only made up about 23% of studies, and in social sciences it wasn't much better, it was about 36%, but slightly higher because they seem to use more of the randomized experiments. But essentially, so we've got quite a big problem here in conservation, is that a lot of studies, a lot of the evidence base is quite methodologically weak, which was quite concerning. So now to move on to the between study biases. And So here I'm again looking at the kind of answering the question of how is evidence distributed in conservation, across the world geographically, but also across species and different taxa. And again, I went back to the conservation evidence database because it's got lots of metadata for all these studies. So I just mainly looked at amphibians and birds. I also did an analysis on primates, but hopefully in the future I'll be able to do a much bigger one because we've expanded the coverage quite a lot to different sort of fields of conservation. And what we found was that again, probably unsurprisingly, the evidence base is really severely biased. And for birds and amphibians, it was very biased towards Western countries. So I'll just give you an example here from amphibians. You can clearly see that studies are really clustered in Europe and North America, and to some extent Australasia. And for birds and amphibians, it's quite a stark difference. So 84 to 90% of studies are actually conducted in North America, Europe, or Australasia. And if we drill down to the country level, 63 to 64% of studies are conducted in the UK, the USA, or Australia. So there's clearly a hell of a lot of bias here towards these specific countries. And actually, if you look at study designs, you also find that the more complex, the less biased study designs are also follow these patterns. So they're also generally quite restricted to these countries and these regions. So we've kind of got a double bias going on there. So the evidence is clustered in, these Western countries, and it's also tends to be of a higher methodological quality, just based on study design. If we move to the taxonomic biases, what we find, also probably unsurprisingly, is that we've got a lot of groups that are missing in the evidence base. So here, just looking at primates, tarsiers, and night monkeys, you can see here their symbols are in white, so they've got no studies in the database. And we also, the colours are meant to represent how over or underrepresented species are relative to the percentage of threatened species they contain. So if they're in dark blue, so the hominidae, that means that's the great apes, so they're very overrepresented relative to how many threatened species are present in that group. And then we've got the colobinae, the leaf-eating monkeys, so like this proboscis monkey, they're very underrepresented. They're quite underrepresented in the evidence base. So clearly, we've got bias going on here between different groups, and it's the same for birds. So you can see these red X's that are sort of around the outside of this phylogenetic tree. Those are showing that there's no studies for those groups. So hornbills and hoopoes and hoatzin, they both have no studies. And also the underrepresented groups here are parrots and songbirds, and the overrepresented groups are shorebirds and waterfowl. And I guess that also kind of follows the biases towards these Western countries where there's been a lot of research on shorebirds and waterfowl traditionally, in the UK and Europe and maybe the US as well. So I think that these obviously these taxonomic biases are also quite linked to the geographic biases. So now I just want to go through kind of my kind of wish list from my thesis as to what we do about these biases. So just to recap, my past research has clearly shown that we've got a methodologically weak evidence base, and we've got severe spatial and taxonomic biases. And I kind of like doing a medical analogy for this, is that if we were in medicine, what this would mean is that we'd have little or no evidence on how to treat patients that, for amphibians and birds at least, that live outside of Western countries and are at greatest risk of dying. And that's clearly not an acceptable situation to be in if we were dealing with humans. And so it shouldn't be acceptable in biodiversity conservation either. And so in my thesis, I tried to structure the last chapter to kind of say, okay, what are we going to do about this? How do we actually fix the evidence base? And so I'm going to go through these 4 themes now and kind of lay out what I think are the ways that we can sort of fix these gaps and biases. So the first of all, I think one major thing that stood out is clearly that study design really matters. And I've got some quotes here. So Light, in quite a nice paper, had this quote: "So you can't fix by analysis what you bungled by design." And I kind of came up with one too, because I thought, why not? So I say study design is to study as foundation is to building. I think that's quite a nice one to tell students when we're teaching them about statistics and study design. So to fix these sort of methodological gaps or biases in the evidence base, we kind of obviously need to facilitate this usage of more rigorous study designs. And one way we can do that is through this idea of carrots and sticks. So my supervisor, Bill, always likes talking in these terms. So carrots to encourage people to use these designs in a nice way is through, things like funding and longer term contracts, so they can do these, maybe more long-term, more complex experiments, more training and education about study designs. One thing I'm amazed by is that if you talk to statisticians, even statisticians don't really learn about study designs that much in undergraduate education. So there seems to be quite a big gap in the training for study design, how to design studies well, even in statistics. So I think we could learn a lot from, or we could even lead the way, I think, in better training, researchers of all ages in how we design studies well, and then we could also think more about what study designs exist from other fields, so like economics and medicine and social sciences. That there are probably designs out there that we can use that maybe we haven't yet come across in conservation and ecology. We also need obviously better links between research and practice, because that will also enable the use of these more rigorous designs. And then to look at the sticks, so how do we sort of more enforce this use of more rigorous designs? Well, I think something that's important to think about is that if there are interventions or impacts we want to monitor that have greater risk, it should really just warrant a higher minimum standard of study design. That should just be part and parcel of what we do. The idea of pre-registration and pre-analysis plans is also an interesting one, and that could help as well, because it's kind of a peer review process before you even do your study. So that could maybe help train, or at least give people better ideas as to how to design their studies well. And I think an important thing to say is that if people do cost assessments of how much a study design will cost to implement, or how feasible it is, what they need to include is also the costs associated with type 1 and type 2 errors from their study design. Because if you misinform a decision maker, or your study is designed badly and therefore it leads to misinformation, that should be a part of your risk assessment or feasibility assessment or cost assessment, because those are important costs to think about. And I think the idea is that also researchers should adjust budgets and project timescales to accommodate these more rigorous designs, rather than the other way around, than trying to fit a study design within a short project. I know that's not always going to be realistic, but I think that needs to be the shift in the culture that we need in research. Okay, so moving on to the gaps and biases in the evidence base. I'm conscious I'm probably running out of time a bit, but so I'll try and go through this as quickly as I can. So I think one way to tackle these gaps and biases that we've got in the scientific evidence base is to actually, do much more of integrating and meaningfully combining evidence from different sources. So that's, looking more carefully at actually, rather than just talking about, oh, we need more traditional and local knowledge in decision making, how do we actually put scientific evidence and this other knowledge together to make a decision? How do they complement each other, and how do we actually have them side by side in decision making? And I think that's quite a big challenge politically, and also just scientifically and in a research sense. We also obviously need a lot more evidence on cost effectiveness, and that's something that a PhD student at in my group is doing. He's called Tom White. So that's quite an exciting area, I think, to build that evidence base as well. Alongside, I guess I've looked at the evidence base on effectiveness, and we also need a lot more evidence on cost effectiveness. And then obviously I've got to talk about Tatsia and Violetta's work on the Translate project, which is amazing. And clearly that non-English literature is really important to filling these gaps and biases in the English literature. So I should point out that all of the research I did was mainly focusing on, well, exclusively focusing on studies published in English. And we've recently been doing more work that Tatsia has been leading on, expanding that into non-English literature. And it's so far, I mean, these figures might be a bit old, but I think last time I checked that they were increasing the taxonomic coverage by, about 47% and the geographic coverage by 7%. So it's really shows that it can be useful, but it is caveated by the fact that I think what was found was that there tended to be less reliable study designs in this non-English literature. So we also have to be a bit careful as to how we then integrate that and how that's, we might fix one bias in terms of the taxonomic and geographic biases, but we might also, obviously maybe worsen or at least not fix another type of bias in the methodological biases. And I think something that's important to remember that sort of I just thought of the other day is that we also really need to think more about how we reverse the transfer of knowledge. So not just getting non-English language studies and putting them into the English literature, but also reversing that. And how do we make sure that decision makers that around the world that don't speak English, how do we make sure they get access to evidence in the language that they use? And just quickly, I think we also need to better prioritize how we gather evidence in the future and how we generate it to fill these gaps and biases. So first off, we need to actually prioritize the actions or interventions we're testing. And this idea of maximizing marginal gains, we should really be focusing on interventions that don't have many studies versus those that have lots. We should focus on the ones that we lack evidence for. That's pretty common sense, I think. But then there's a question as to whether we focus on interventions with a few studies versus those with no studies at all. And I think actually we might be better focusing on those that have some studies rather than no studies, because we can actually raise the evidence, the kind of level of evidence in interventions that have a few studies to a more acceptable level to make decisions than we can if we focus on those with no studies. So, and then afterwards, we can then shift to those that don't have any evidence. So the idea of this is that you build up lots of more interventions or actions that have got a stronger evidence base. You might be able to do fewer of them rather than having you're spreading the evidence thinly across lots of interventions, and you have lots of interventions with very little evidence. So this is just one idea as a strategy of how to go about this. And I think obviously it can't just be about the amount of evidence that each study, each action has at the moment. We also obviously need to focus on probably interventions that are highly costly or risky, and also those that are commonly used or implemented, because I think these represent a potentially large waste of resources and conservation if we then show that those interventions are ineffective. So those are the ones I think we need to target, and obviously target those that are designed to conserve threatened species and habitats. And then once we've obviously prioritized the interventions or actions we're going to test, we then need to prioritize the studies that we're going to use to generate the evidence. And you can do this in two ways. You could prioritize quality or coverage, i.e. quantity. And you could also think about whether you're focusing on reproducibility or generalizability. So do you want lots of studies in the same context so we can test whether results are reproducible, or do we want to actually test studies in lots of different contexts so we can see whether the results generalize across different locations and species? So that's also another question we need to think about. And I would say, clearly based on my previous work, if we've got riskier interventions that we're testing, we really need to prioritize doing more reliably designed studies, because it's important that we get those results right. And for the less risky ones, maybe it's more sensible to prioritize expanding the evidence base, the coverage of the evidence base, at least in the short term. And we can also think about the species we prioritize, whether they're threatened. Obviously, that would be ideal, and maybe focusing on the tropics as a way of starting to fill those gaps. And another interesting thought I had was that, should we actively discourage the testing of interventions in well-studied regions? So should we actually try and divert resources from, like, North America, Europe and Australasia to these other regions? And so really shifting the research effort sort of in a more sort of direct way. And so there's lots of ways we can do this. I'm going to skip over this, but I think it's important that we're mobilizing that evidence from underrepresented locations, but without falling into the trap of helicopter or parachute science. So we're building local capacity to do that research, and we're not going in as Western researchers and doing the work for them. That's not the way it should be done. And one thing I find quite interesting is, why don't we already have some kind of approach or organized approach like this to testing conservation actions or interventions and solutions? Why don't we have some prioritization like we do for maybe other issues? Do we need some sort of organization or fund that kind of takes charge of this? So those are all open to discussion, I guess. I'm going to skip over this because I think I'm running over a bit. But essentially here I was talking about the need to kind of speed up the process of evidence synthesis as well, because the amount of studies that are being published now that we need to sort through to get the evidence is really increasing and accelerating dramatically. So we need cool things like automation and machine learning to help us synthesize the evidence more quickly. And I think we also need to shift in how we think about evidence and giving evidence to decision makers. So moving from kind of what works generally to actually what works here for a given decision maker. So what works for their local context. And trying to answer, this is something I'd like to do in my fellowship, is answer whether certain interventions or actions are likely to be, universally effective. Do some things just work everywhere? Are they effective at achieving desired outcomes everywhere? Or are there lots of interventions that are just very context specific and dependent? And then can we give recommendations as to where they work and where they don't work? And finally, just to say, building on this, we really need to promote embedding evidence use in practice and policy a lot more. And I think the key to doing this is providing locally relevant evidence-based recommendations rather than generic ones that are often given in systematic reviews and other forms of evidence synthesis. So one way I'm doing this is through this evidence to decision tool, which guides practitioners through this process of making an evidence-based decision and laying out the steps and the evidence that they've used to come to a decision, and then transparently reporting that in a kind of document at the end. And crucially, what this does is it tells them to, shows them to get evidence from scientific literature, but also from local knowledge, and look at different aspects of the effectiveness and other aspects of an intervention that are desirable, like its cost effectiveness, its acceptability to stakeholders, its feasibility. And we've got the evidence champion schemes as well at Conservation Evidence. So we're doing a lot of work to do workshops and events that show them how to use these kind of decision tools, and also how to generally just use more evidence in their day-to-day decision making. And just finally, we're also, some of my work that was in my postdoc since March is looking at designing sort of dynamic and interactive interfaces into meta-analyses. So can we provide a kind of this online website called Metadataset, can we provide a way for decision makers and users to actually go in and get customized results from meta-analyses based on what they're interested in, where they work in the world. So that's kind of the idea with that, and trying to tailor these kind of evidence syntheses and products to the users' questions of interest. So yeah, and I'll just quickly go to my take-home messages. So clearly what I've shown here is that scientific evidence in conservation has many and often severe gaps and biases. And we obviously need to fix those urgently. And the way we need to do that is through more rigorous testing of actions, better prioritize the actions we test and the evidence we generate, embrace new ways of collating evidence, combine these different sources of evidence to fill these gaps, so non-English and English literature, scientific and local knowledge, and crucially kind of assess that local relevance of evidence to different decision makers, so making sure we tailor our recommendations to their local context. And I think also to embed evidence use, we also need to co-design and build tools like the one I briefly showed before to help decision makers better use evidence. Okay, that's my whistle-stop tour of my research. Thank you for listening. Any questions, I'll be happy to answer them.